So second block, we're going to talk about Golden Gate assembly, guys. And this is a really great technique, just period, great technique. If for some reason you didn't catch the first block, if you're watching this later, we just talked about primer design. This is Golden Gate assembly. My name is Dennis Mishler. I'm a professor at UT Austin. Okay. So Golden Gate assembly, it works really efficiently with standardization in parts. In synthetic biology, we have a big emphasis on standardized parts, systems, and moving forward. And Golden Gate assembly is that exactly. It's really nice in that regard. It was first introduced in 2008, so it's younger probably than everyone watching this, unless we have a couple of 12-year-olds, right? So it's a relatively new technology. Many pieces of DNA can be assembled in a single pot with Golden Gate assembly, and you can combine not just multiple pieces of DNA, but you also combine the digestion and the ligation steps into a single reaction. All right, it's all in one tube, just goes. Right. The key to these, to the Golden Gate assembly technique, though, is type 2S restriction enzymes, and I'm going to talk about these in the future slides. Okay. So, Golden Gate assembly. Here's a little bit of an overview of it. This is going to be, if you haven't seen it before, guys, this is going to look a little daunting, and I apologize for that, but I think hopefully it'll be okay after a little bit. So there's a few different steps to Golden Gate. I'm gonna get my pointer going again. Boom. There's a few different steps in Golden Gate assembly on this design here. Step one, take a DNA sequence and you got to make it into something we call a part plasmid. Okay. So you take your DNA sequence. This could be a PCR product. It could be a synthesized gene. You have to put it into an entry vector. You do the reaction. Boom. You get a part plasmid that looks like this. Step two, you can take lots of part plasmids, several of them all at once, put them into a single reaction together, and you get what's called a first stage plasmid or an assembly plasmid here. And you can see we have this green, orange, and red parts, which were here, here, and here, right? And you can assemble that all together. And then what's even cooler is you could take multiple first stage assemblies shown here, and you could combine them together to get this, the second stage assembly. Okay. And so ultimately you can put a whole lot of DNA together into single plasmids and then take those single plasmids and move forward to make more complex plasmids. And it's done really quickly. Okay. So type 2S enzymes, this is really the big thing here. And let's go through a little bit of sort of the older school traditional restriction enzymes people would use. And then we'll talk about the type 2S. So this is a type 2 enzyme over here. This is EchoR1, that's one example. And here's our DNA sequence. And you can see the type 2 restriction enzyme recognizes and cuts this palindromic sequence. Okay, the scissors indicate where it's going to be cut, and the purple part indicates the DNA that the enzyme actually recognizes or binds to. Okay. So EchoR1 here, the protein is present, and it's going to cut there, right? So you have to cut the DNA exactly where the restriction site is, and you have to stitch it together again to do anything. Type 2S, though, is very different. Type 2S, here's the DNA sequence. Notice that there's a sequence here, and there's a bunch of ends. That's really important here. The BSA1 enzyme is one example of a type 2S restriction enzyme. And here you can see it has a recognition site where it binds or recognizes. And then the enzyme actually cuts adjacent to the recognition site. And it's cutting at all of these ends here. In DNA, when we refer to something as being an N nucleotide or an N base, that means it could be anything. It could be an A, a C, a G, or T, whatever. So this could be ACGT. This could be CGGA, whatever you want. And that's really powerful because now what happens is we can cut at this sequence and we can stitch it back together. And we don't even care about this over here. We can get rid of that, right? We can get rid of this cut site, or not cut site. We can get rid of the recognition site. And so we can make our assembly really powerful. So we cut with our type 2S enzyme here. And what will happen is we drop out the recognition site, we get rid of that, and we just keep our part, okay, our DNA sequence here, and we can stitch it all together, right? And so type 2S enzymes allow us to do seamless or scarless cloning, all right? We can get rid of any sequence we don't want, and we can keep just the sequence we really need. You can vary the cut site nucleotides, so these can be anything you want, allowing you to generate multiple different overhangs in a single reaction, right? You could add a whole bunch of different parts with different overhang sequences, and this would allow you to assemble many pieces of DNA together in a predictable order, right? You could have 4, 5, 6, 7, 8 parts, and if you design it correctly, they'll all stitch together in the right order, okay? So here's a little bit more detailed example. This is one of the slides I grabbed from Renee, which I thought was really, it's a great slide. So here we're going to start, this is our part plasmid over here. This symbology, this little symbol here, this is a connector symbol right here, okay? And you can see this part is the connector. Before and after the connector are the overhangs, right? So this is an overhang here. This is a different overhang sequence here. And what we can do is we can combine all 8 of these parts into a single reaction, do the assembly, and now we get this great plasmid. And notice it has all these different overhang sequences that come together to ligate, right? All of these are different. They all differ by at least 2 nucleotides. That's beautiful, right? It's awesome. And then what you can do is you can then take this is called the transcriptional unit commonly, not always, but commonly. You can take this transcriptional unit and combine a whole bunch of them together. 1, 2, 3, 4, 5 TUs. Wow. So 5 TUs, each TU had 8 parts together. Wow, that's like 40 different part plasmids were combined to get to this one large plasmid at the end. Okay, that's really awesome. That's really powerful. It really uses standardized parts, right? So it's really nice. Okay, so now we have all these standardized parts. That's really important. And so you need part collections. And so this is one set from Lee Duber from 2015, where they have 8 different part types, and they have a whole bunch of different parts for each type. So if you worked within their system, you could easily interchange the one part type 3, one coding sequence with another, or one promoter with another, and everything would work together still. Okay, so it's really nice. The key is you have to design it well. All right, you have to design it so that the overhangs are properly complementary. This leads to modular cloning. Renee is going to talk more about that later. I want to highlight that the most common type 2 enzymes are BSMB1, BSA1, and sometimes VBS1. Some though are being replaced with more efficient or better enzymes, especially NEB is pushing EPS31 as a better enzyme than BSMB1. I'm not going to go into those details, but there are newer, better enzymes coming out for these all the time. And so the process is just becoming more efficient. Okay. Many web apps also have assembly wizards, which will help you design these. And that's really key, I think. So, you know, that's a great thing. And we'll have enough time here to go through that a little bit with me, with Benchling, and then Renee will go through it more with his, I think Genius is his program. Okay. So Golden Gate in your lab. Once you've done all the design, once you've done all the stuff where you've made your sequences, and then the DNA sequences arrive in your lab, it's pretty easy. You take your part plasmids or your DNA sequences, you put it into a single reaction. You put that reaction in a thermocycler, right? A thermocycler, although we call it a PCR machine sometimes, a thermocycler just changes temperature. And so you run a procedure similar to this. This one goes for 6 hours. I've seen some that are faster, but this one's fine. You just run between 2 different temperatures back and forth. The 37 degrees Celsius allows the restriction enzyme to activate, cutting DNA, cut. The lower temperature then gets to a ligation step, allowing the ligase to then put together cut pieces of DNA. And you just cycle back and forth a whole bunch. And so while you start with a bunch of your different plasmids that are separate, and you start with none of your final assembly, over time, as it goes along, you end up with a lot of your assembly and hopefully none of your original parts. Okay. Once that reaction is done, you just put it straight into competent cells. You do a transformation. So we're going to talk more about transformations in webinar 6B in a couple weeks. But basically, you just put them into your cells. You transform overnight. Next day, you can come in and see if it worked. Okay. So it's a real quick, fast procedure. Some of the key takeaways are right here, guys. DNA pieces are combined using the type 2S restriction enzymes. Each part type has a specific prefix and suffix sequence, which includes that overhang. This allows you to dictate the sequential ordering of your DNA sequences. And the single reaction includes both the digestion and ligation steps. You don't need to purify your DNA or do gel extraction, right? If you do BioBrick cloning, for example, you really need to gel extract. You don't need to do a lot of these steps. You just do it all at once. The advantage is many pieces of DNA can be assembled really, really quickly, really, really fast. The disadvantage is that it's a lot more difficult to do initial planning and to initially figure out how the system works. But once you get over that initial hurdle, it's real fast, okay? So we should have enough time here. I think we do. Yeah, we have enough time here. So we're going to go ahead and do a little bit of Golden Gate assembly using Benchling. Again, I have these two sequences here. You guys can look at them later. I'm not going to post them in the chat right now, but the same thing applies. You can copy these in Benchling and then potentially look at them if you want. So we're going to quickly do this. Let me go ahead and do my screen share switch over. And so for this, I'm going to use two sequences here. I have a PCR product for iGEM demo. I love making PCR product sequences. Even before I do PCR, when I'm designing my primers, I always make PCR product sequences so I know everything looks okay. And then we're going to have to end up also using this PYTK sequence. I'll get that up there. So here we have our primers that were used to make this sequence. Notice it has a BSA1 cut site and it has a BSMB1 cut site. The purplish blue box is the recognition site, and the black line is the cut site. So I'm going to assemble with BSMB1 because I have one on the forward primer area and I have one over here. Okay. So I'm going to go to the assembly wizard. I'm going to create a new assembly. I'm going to do Golden Gate assembly here. You can see it has some details. I'm going to go ahead and click start. And now I need to set a backbone and insert fragment. This is my insert sequence. I want to make this into a part plasmid. So I'm going to click insert. And now I need to use some BSMB1 cut sites. And I need to use the ones that are in between, in the middle. So I'm going to go to here. I have to use the 12 position, which is up here. And I need to use the 701 position, which is here. So that's going to be my insert sequence. I'm going to keep that. I'm going to set that fragment. Boom. It says this is what I'm doing. And now I'm going to click on the backbone tab. And I'm going to go to my backbone sequence, which is this guy here. And I need to set the fragment for this one as well. And I'm cutting with BSMB1. Now, if I wasn't cutting with BSMB1, I can change this. I can go over here and click on BSMB1. I can choose a different one if I wanted to, all right? So if you wanted to cut with BSA1 or something, you could do that instead. But BSMB1 is good for me. I, again, need to set my backbone cut sites. So here's my first one. And you know what? The funny thing about this guy is I actually want to take this part of the sequence and then this part of the sequence down here. So I have to cut starting with this bottom one on this system. So I need to go with this one first, which is around 1040. So I want to do 1046, great. And then I need to go all the way down here, back up, because this is a circular plasmid. This is a circular plasmid. I need to go to this guy here. So I'm gonna go to 16. And now between 1046 to 16, that's gonna be my second fragment. So fragment 1, fragment 2. Overall assembly looks like everything checks out. So Benchling has done some double checking for me to make sure all the cut sites, all the overhangs look correct. I'm going to go ahead and just call this demo 2. Okay, I'm going to click assemble. Just ask me where I want to save it. That's fine. I don't care. Okay, and now we have a plasmid assembled. Okay, so let me go ahead and take a look here. Let's look at the plasmid map. You can see a green piece and an orange piece exist. The orange is the backbone sequence from before, and the green piece is the insert sequence. Okay, so it's color-coded it, so I know where everything came from. And if I look at the linear map, it looks the same. And if I look at the sequence map here, I can actually, if I scroll down, I can find where it was stitched together. Okay, so it highlights for me the stitching, and I can see I still have my BSA1 cut site there. So I could use this part plasmid in another assembly later with BSA1 to now put together a bunch of part plasmids. Okay, so pretty quick and easy, right? It sounds a little scary at first. It's a little different, but you can get through it pretty well. Okay, so with that, again, you can look back at the webinar. It's going to go online in the next couple of days, and you should be good to go. So hopefully you learned a little bit about Golden Gate assembly here. It takes a lot of practice to master it, and Renee is going to go over some more stuff with you. And with that, if we have questions, let's go ahead and answer questions, guys. And I'll give you guys a couple of minutes, of course, to think about questions or let me know. And I know we go through these things fast, but unfortunately, with short periods of time, we just have to.
And I mean, all the videos will be uploaded, and you can click and stop every video in exactly your pace. to learn everything slowly.
Any questions, guys?
We continue.
Feel free to ask, guys. Don't worry. A simple question is a great question. I'm not gonna be upset. You can always let us know. Has anybody used Golden Gate Assembly before, guys? No.
But that's good. I mean, that's really good, because then this webinar is perfectly for you.
Someone in capitals was like, no. Yeah. So no worries. This is a lot. When I teach Golden Gate Assembly to my students during the academic year, guys, we do this several weeks in a row. We go over it, and then we go over it again, and we go over it again. So,
yeah.
Do you recommend? Oh, here we go. Great. Do you recommend we use Golden Gate if our advisors have no experience using it? It's going to depend a little bit on what your goals are. If you're trying to work with a lot of plasmids, I would say yes. And I would say, advisor, I'm going to bring you into the 21st century, and let's do it. If you're only going to do a little bit of cloning, it may not be worth the effort. But here's what I'll tell you. If you're going to do digestion-based cloning, it is the best way to go about it. Okay. Now, somebody said, why would you use Golden Gate over Gibson? So here's why. Gibson doesn't really allow you to scale up as much in terms of parts. Like Gibson allows you to make a part. So maybe if you want to make a first part plasmid, you use Gibson. But later, if you want to put a whole bunch of stuff together, I think Golden Gate Assembly gives you a lot of interchangeability. You can move things around very easily. And so if I wanted to try out 2 or 3 promoters or coding sequences or something like that, having them all as parts in Golden Gate Assembly, I can now mix and match those all together really quickly. And so it scales really well. Gibson is great if you're doing smaller, like if you're doing one thing you have to make, that can be great. But they're different, and they both have different times when they're very useful, okay? And we'll do Gibson assembly, I think is next week maybe. We're gonna have some people talk about Gibson and yeast assembly next week. Somebody asked about the video. It's on the iGEM measurement page. So if you type in iGEM 2020 measurement, you'll see there's a webinar page that has a lot of materials up there, including the slides as well as the videos. We did that one. What level do your students have when you teach them this? Freshmen. Yep, freshmen. So my students in the spring are freshmen. My students in the fall are sophomores. I also have more senior students who help me teach and go through things. But this spring, since everything was remote for, let's say, a month and a half of the course, we basically did primer design, Golden Gate assembly, making PCR products, DNA sequencing analysis, all on with our freshmen. And so we created modules like this, and we created videos and such. I thought about sharing some of those with you guys, but they use a lot of DNA sequences that are very specific to our research, and I don't know that it would be easily applicable to what you guys are all doing. Okay, so real quick, here's a couple other points. Primers differ. Do you design primers differently with Golden Gate assembly versus Gibson? Oof. The answer is going to be kind of yes. I don't want to get into those details because I don't do a lot of Gibson, but that's a great question for next week. Sonia, I think, is the one leading that webinar, and she's awesome. So you can ask her that question. Backbones don't work with every organism. That's correct. Yeah. So good point. So different organisms need different backbones or origins of replication. That's the part that normally matters the most. And so you have to have a part plasmid if you're using Golden Gate assembly. You need a part plasmid that has an origin that is compatible with your organism.
Yep.
So the reason I used the Lee Duber example in the webinar today is because they actually have a yeast and E. coli origin, because they're doing assembly both. They're doing assembly both ways. They want to be able to go to yeast or to E. coli, and so they have both origins present. So you could do something like that, right? You could clone originally in E. coli, and then on the last step, add in something else to go to another organism if you wanted to. You know, if I missed somebody else's question, I apologize, but I think I got all of them, and I do want to make sure that Renee doesn't feel like he's crunched for time. So thank you guys. If you have more questions, again, reach us at this email address down here. You can take a look at the sequences, and again, the webinar stuff will all be online within the next day or two. Okay. So with that, Renee, I'm going to go ahead and stop my sharing, and you can start screen sharing